Greetings from Everyday GMP. I am your presenter from Everyday GMP. This is series of video. We will discuss all the definitions and terms used in EU guidelines for good manufacturing practice for medicinal products for human and veterinary use. In this video we will cover terms starts from alphabet A and B. Further series will be covered in other videos series of definitions. Video number 1, keep watching all videos. So let's start. In the world of pharmaceuticals, precision and adherence to regulations are paramount. Understanding the key definitions and terminology laid out in the European Union Good Manufacturing Practice Guidelines, Annex 1, is not just important, it's essential. Join us on a journey through the intricacies of pharmaceutical manufacturing as we dive deep into the terminology that shapes this industry. In this comprehensive video series, we will demystify the definitions used in the EU GMP Guidelines Annex 1, breaking down complex concepts into easily digestible explanations. Exploring pharmaceutical definitions from EU GMP Guidelines Annex 1, unlock the language of pharmaceuticals and dive into the core terminology that shapes the industry. Join us on this educational journey to understand the key definitions that ensure precision and quality in pharmaceutical manufacturing. Subscribe and let's begin. So before start, subscribe the channel and press the bell icon. So let's start. Before we start, please subscribe to get more informative videos. So, let's start. Airlock, an enclosed space with interlocked doors, constructed to maintain air pressure control between adjoining rooms, generally with different air cleanliness standards. The intent of an airlock is to preclude ingress of particle matter and microorganism contamination from a lesser controlled area. Look at image, it will be more cleared. Water action limit, an established relevant measure, e.g. microbial or airborne particle limits, that, when exceeded, should trigger appropriate investigation and corrective action based on the investigation. Alert level, an established relevant measure, e.g., microbial or airborne particle levels, giving early warning of potential drift from normal operating conditions and validated state, which does not necessarily give grounds for corrective action but triggers appropriate scrutiny and follow-up to address the potential problem. Alert levels are established based on routine and qualification trend data and are periodically reviewed. The alert level can be based on a number of parameters including adverse trends, individual excursions above a set limit and repeat events. Aseptic preparation or processing, the handling of sterile product, containers and/or devices in a controlled environment in which the air supply, materials and personnel are regulated to prevent microbial, endotoxin, pyrogen and particle contamination. Aseptic process simulation, APS, or media fill run. A simulation of the entire aseptic manufacturing process in order to verify the capability of the process to assure product sterility. Includes all aseptic operations associated with routine manufacturing, e.g. equipment assembly, formulation, filling, lyophilization and sealing processes, as necessary. Asepsis, a state of control attained by using an aseptic work area and performing activities in a manner that precludes microbial contamination of the exposed sterile product. Bacterial retention testing. This test is performed to validate that a filter can remove bacteria from a gas or liquid. The test is usually performed using a standard organism, such as Brevimontanus diminutor, at a minimum concentration of 107 colony-forming units per square centimeter. Barrier or RABS or isolators. There are a physical partition that affords aseptic processing area, usually grade A, protection by separating it from the background environment. Such systems frequently use in part or totally the barrier technologies known as RABS or isolators. Bioburden: the total number of microorganisms associated with a specific item such as personnel, manufacturing environments, air and surfaces, equipment, product packaging, raw materials, including water, in-process materials, or finished products. Biodecontamination, a process that eliminates viable bioburden via use of sporicidal chemical agents. Biological indicators, a population of microorganisms inoculated onto a suitable medium, e.g., solution, container or closure, and placed within a sterilizer or load or room locations to determine the sterilization or disinfection cycle efficacy of a physical or chemical process. The challenge microorganism is selected and validated based upon its resistance to the given process. Incoming lot D value, microbiological count and purity define the quality of the Blow Fill Seal, BFS. A technology in which containers are formed from a thermoplastic granulate, filled with product, and then sealed in a continuous, integrated, automatic operation. The two most common types of BFS machines are the shuttle type, with parison cut, and the rotary type, closed parison. That's all for today's video on terms and definitions used in pharmaceuticals. If you found this information helpful, give us a thumbs up, subscribe to our channel, and hit the notification bell to stay updated on more job, immigration and educational content. If you have any questions or want to share your journey, leave a comment below. Until next time, keep dreaming big and reaching for your goals.